Hi, this is Dr. Wang. I'm providing a recording for the practice exam for midterm bioinfo. This practice version is very similar to the actual exam. So let's look at the first question. Here we have a GenBank ID. So I'm going to copy the ID and then open the GenBank, paste my ID in there, and then search. So the first question asks for species. So that information is located under source species name right here for question 1. So for question 2, we are in here actually looking at the same GenBank record. I do want to emphasize that during the actual exam, your question 2 will have a different GenBank ID. So please do another search to pull out the record. All right, so for this practice, we're simply going to look at the same ID, and then this asks for how many exons, right? So in here, this information is under features. So under features, one of those display will be exon. So I'm going to click on exon. This opens up an information tab at the bottom of the page. As I flip through this, you can see different regions of the gene are highlighted. So we're looking at exon 5, exon 6, exon 7, 8, etc. And then there are a total of 11 exons, right? So this is displaying how many exons there are. So this is question number 2. So for question number 3, asking for how many chromosomes. Again, in the actual exam, please do the GenBank search one more time before you pull out this information. So in here, I'm still going to go to the same GenBank record, and the chromosome information is located under features, source, chromosome. So chromosome 6, just enter 6 for question 3. So question 4, so how many base pairs? This information is located right at the beginning and the locus. So we can see that there are a total of this many base pairs. So simply enter this number into the box, right? So please do not enter BP. You are just going to enter an integer. Okay, so question number 5. So here, this is actually a BLAST search. So I'm going to copy this entire sequence and then go to BLAST. Since this is a DNA sequence, I'm going to do a nucleotide BLAST, paste the sequence in here, use the default setting, make sure that you are searching against the entire nucleotide collection. So BLAST. I do have the BLAST result already. Oh, actually, it's coming up quickly this time. Okay, so we can see usually the very first hit is the record that's pulled by BLAST. So as you can see here, this is 100%. So I'm going to click on this. So over here, you can see that this is 100% match, right? So for this particular record, the species name is actually in the title. It's not always the case. However, you can always click on the GenBank ID that's right underneath to go to this record and then go to source, which will definitely list the species name. So this is for question 5. So for question 6, we're going to look for the longest open reading frame. So I'm going to copy this sequence, go to Sapio, open a new plasmid editor, and then paste the sequence in here. So we're going to look for the longest open reading frame. So go to translation, ORF detection. Make sure you start from the very beginning and then just scan. So after scan, you are going to see that 2 open reading frames were identified. So this one and then this one. So the longest open reading frame, certainly this one looks longer, right? So, and it's when you put your cursor over the green line, it's also telling you that this one has 210 amino acid. If you are not, if you want to be absolutely sure, you can certainly click on the green line to select the entire region and then go back and then translate. This is on the forward direction because it's going to the right. So, and this is also going to tell you there are 210 amino acids. So this is the answer for question 6. So question number 7, ask for, so if you use this as the template and the 2 primers below, what is the length of the PCR product? So in the actual exam, this sequence is going to, again, differ from question to question. So a safe way to do this would be, I'm going to copy this sequence. All right, so, and then I'm simply going to open a new plasmid editor and then paste my sequence in here. All right, so the next thing I'm going to do is to find the primer. So I'm going to first copy the sequence for primer 1. All right, so make sure that you copy the sequence only. Please do not copy 5' dash or any space in there. It's sequence only. This is primer 1. So go to annotate PCR primer, paste the sequence in here, and then click the search button. As you can see, a match discovered, and this is on the forward direction. So I'm going to call this primer 1 and then create. Coming back to the question, copy primer 2 sequence. All right, so go again, annotate PCR primer, paste the primer sequence in here, and then search. There is no match, which means that this primer is on the reverse direction. So I'm going to change this to the reverse direction. When I change this to reverse, the sequence here actually changed. So let me make sure that I paste primer 2 sequence back in there one more time and then search. This time there is a match. So here, this is our primer 2. Create. All right, so we can see on the graphical form, we have primer 1 and primer 2 running towards each other. So this is going to work in PCR. To simulate the PCR, click on this button over here to look at both primers. Select primer 1, hold Shift key. While holding the Shift key, click on primer 2. So when both primers are selected, click on PCR product. This simulates the PCR reaction. So we can see that this product is 433 base pair long. So this is how to solve question 7. Question 8. So if you digest this in this sequence with NLA3, how many fragments are you going to get? So here, again, I'm going to copy this sequence, make sure that I open a new plasmid editor window. All right, so this is really important for the actual exam because sequence differ from question to question. Okay, so now we are going to do a simulated digestion with enzyme NLA3, Roman numeral 3. So click this button to look at all the restriction enzyme. Go to select list, make sure NLA3 is actually selected. Okay, so let me go fast forward. Oh, I might have passed. Okay, NLA3, check, make sure this enzyme is selected, apply, and then close. All right, so now we are ready to do the actual simulated digestion. So go to analysis, click restriction analysis. So over here, we're going to, currently we only have this one sequence here. So I'm going to select, right? Since I opened a new plasmid editor, so I know for sure that this is the sequence that I was looking at earlier. So click the sequence and then select NLA3. So let me, NLA3 right here. All right, so we can see that NLA3 cuts multiple times, generating a total of 1, 2, 3, 4, 5 different fragments. And then the longest fragment is right here, 585, right? So those are basically how many base pair for those 5 fragments. So this question asks for how many fragments, right? So there were 5, and then the longest of them is basically the longest over here. And I have to emphasize again, in the actual exam, the sequence in question 8 and question 9 will be different. The enzyme name might differ as well. So please make sure that for each question, you start a new plasmid editor. So you're starting from a fresh. So question 10. This is a sequence alignment question. So the first thing I'm going to do is to copy the wild type protein sequence, right? This is not DNA sequence, it's protein sequence. So I'm going to copy that. And then over here, I'm going to open a sequence alignment tab. So paste the protein sequence in here. I'm going to call this the wild type. Go back to the question, copy the mutant protein sequence, and then paste it into the second window, and then I'm going to call this the mutant. Since we are lining up protein sequences, make sure that under sequence type, we choose protein and then run analysis. All right, so over here, we can see that here is this one difference between the two. The wild type amino acid is a T, it's at location 60, and then the corresponding mutant amino acid is a A, it's an alanine. So the answer for this question would be starting with the amino acid in the wild type sequence. The number is the position of this amino acid, followed by the amino acid in the mutant. So this is the answer for question 10. All right, so question 11. Line up the two sequence. All right, so and then distinguish wild type and mutant sequence. So for this one, we actually don't really need to line up. We can do a restriction analysis right away. So here's how to do it. First, I'm going to copy the wild type sequence. All right, so go to plasmid editor. Right, so here, make sure open a new plasmid editor. I'm going to put the sequence in here. Okay. Right. So over here, I'm going to call this the wild type sequence. Right. So because I just copied the wild type sequence, and then I'm going to copy the mutant sequence. Open another tab. Paste mutant sequence right here. Click on the information tab to change the name to mutant. Right. Yeah. All right. So now we can actually do the restriction, simulated restriction digestion, right? So we have 5 enzymes. So the first thing is to make sure that all 5 enzymes are selected on the enzyme list. So I'm going to go to the enzyme, select list. So this opens up the list. So now I'm going to actually pull this a little bit to the side so that I can look at all the enzymes, right? So here are all the enzymes. I'm going to, pull my Sapio a little bit to the side as well, so that I can look at the enzyme and make my selection. So the first one we're going to select is APA1. So, okay, so this might take some time. APA1 is right here. All right, so the next one, BPU14. So BPU14 1. All right, so this is not selected, so I'm going to make sure it's selected. The next one, BSM1, so it should be fairly close to this. Right here is BS, okay, BSM1. All right, so make sure that this is selected. And then I'm going to go to the next enzyme, which is Hindi 3. Hindi 3 is already selected. And then the last one is PAU1. Okay, PAU, right here. So PAU1, okay, I'm going to apply. All right, so here I'm going to now close and then close. So now with all 5 enzymes selected, we can actually start the analysis. I'm going to click on this tab, go to restriction analysis. This opens up a new tab. I will select my wild type sequence and my mutant sequence. And this is why it's important to open up a new plasmid editor for each question. This way, when you do analysis, then the wild type, instead of having a bunch of sequences from all the previous questions, you're only going to have this 2 that you need to work with. All right, so now let's scroll down. We are going to select enzyme, right? So we're going to do APA1 right here. So as you can see, wild type mutant looks similar, right? So therefore, this is not the enzyme to distinguish wild type from mutant. The second one to test is BPU141, also very similar. Okay, so now let's go to BSM1. It did cut, however, the pattern are the same between wild type and mutant, so not this one either. The next one to test is Hindi 3, which is right here. Okay, so now we can see that there is a difference. Wild type, there are 2 fragments, mutant, there's 1 fragment. So Hindi 3 is the answer. I can certainly go to PAU1, as you can see, no difference, right? So this is how to solve question 11. So question 12, we are going to pick a pair of primer that will allow you to amplify a region containing this mutation. So the first thing to do is to copy these sequences. So I'm going to copy and then make sure that I open up a new plasmid editor for question 12. Put the sequence in here. Okay, right, so here is my sequence. The next thing I'm going to do is to locate primer ABC onto the sequence. So make sure that you copy the sequence only, right? So for primer A, and then annotate PCR primer, paste sequence in here and the sequence, and then hit the search button. There is no match, meaning that this is on the reverse direction. So I'm going to change it to reverse. Delete the sequence that is in here and then paste primer A sequence back in there one more time. This is really important, right? So make sure that you paste primer A. All right, so here, as you can see, a match discovered. So this is primer A. I'm going to hit create primer A right here. So primer B, all right, so we're just going to do the same thing for primer B. Annotate PCR primer, paste primer B sequence in here, click search. Actually, there is a match on the forward direction. So primer B is a forward primer, right? So primer B, create. This is primer B, a forward primer going to the right. So now let's do primer C. Copy primer C sequence, coming to annotate PCR primer, paste the sequence in here, hit the search button. There is no match, meaning this is on the reverse direction. So here I'm going to delete the sequence that's in there, make sure I paste primer C back in there, and then search. Now there is a match. So here, primer C, and then create. All right, so primer D, all right, so copy, coming back to annotate PCR primer, paste sequence, search. There is a match, right? So here, primer D. Primer D. Okay, so in this entire process, before hit create, please hit the search button first. If there is a match, and then create, right? So otherwise there might be some mistake, right? So make sure that you can find the match before hit create. So over here, all the primers were discovered on the sequence. I'm actually going to scroll through them to make sure that they look okay, right? Primer D, primer A, yeah, looks okay. And then primer B, yeah, looks normal. And then primer C is right here. Looks normal as well. So which pair is going to work in PCR? I'm going to actually look at the graphic version of this. So this is the entire linear sequence, right? So this is actually another important thing that I should have mentioned when I talked about question, let me see, restriction, right? So it's question 9, right? So question 9 and question 8, this is really important, is that you have to make sure that your sequence is linear, because linear is the default format for DNA. If we don't say anything in the question, then this is a linear DNA. If it's a circular DNA, we would have told you this is a circular DNA. This is linear. So especially for simulated restriction digestion, it's going to make a difference. So please make sure that you select linear for all your sequences, unless we tell you it's circular. Right, so sort of a little side track. So coming back to question 12, right? So we have the 4 primers already identified. Which pair is going to work for PCR? We need to identify a pair that are running towards each other, such as primer C and primer B, right? So we can see primer is running to the left, primer B is running to the right, so they're running towards each other. A pair like so will allow you to amplify a PCR product, right? So looking like this. And there is another option, so which is primer D. Primer D is also a forward primer. So primer D and primer C is going to work in PCR as well. So this is the only 2 options, right? So primer A is right here. You can see this is a reverse primer running towards the left. There's really no other primer that can pair up with primer A. So we have 2 options, primer B, B and C versus D and C. So which one is the correct answer? For that, we have to look at where the mutation is. The mutation is in location 54. 54 is roughly, right, so over here, I can put my cursor and it's going to tell me where my cursor is, right? So 54, I would say it's somewhere in this region, right? Somewhere in here. This is where the mutation is. So you can see that primer D and C will allow you to amplify this mutation, right? So 54, this location is right in the middle of this PCR product. However, primer B and C is not going to work, right? So mutation is right here, B and C, PCR product is right here. So B and C are not going to work. So to solve question 12, locate all the primers first, then pick a pair of primer that's running towards each other with the mutation that's located right in the middle of this PCR product. So that's how to solve question 12.